Hello everyone! So in the last video we discussed experiments, what we learned from these experiments, chromosomes, and sex chromosomes in specific. And in this video I wanted to continue to dive into this topic of chromosomes, and I want to explore linked genes, recombinants, and abnormalities that we find in the genes. So let's get started. So up until now we have talked about people obtaining genes and how these genes are inherited. But there is also the concept of linked genes. So remember in mitosis when we talked about how it goes through two phases of PMAT, and how in the first phase the homologous chromosomes pair and complete PMAT, and in the second phase the single chromosome is separated and four daughter cells formed? Well, in the first prophase phase of meiosis, there is a process called crossing over that occurs. Crossing over is when part of the chromosome on the homologous chromosomes switch places. This allows the daughter cells that form to be different than the parent chromosomes, as if this didn't occur, then just the same genes would be replicating and they would just result in four daughter cells that are basically just fragments of the same parent genes. But this crossing over allows both the mother and father's genes to be on the same chromosome, and for the offspring to express both of these traits. So, and this crossing over is possible because of linked genes. So linked genes are genes located in the same area of a chromosome that are passed on to the offspring together. This switching and the offspring being different is called recombinant chromosomes because they do not look exactly like their parents. So scientists can actually calculate the frequency at which two specific genes will end up on the same chromosome after crossing over. So they suspected that the greater the distance between the two genes there was, the more likely the crossing over would occur between these, and thus they would end up on different chromosomes. Also, if the scientists are just given the recombinant frequencies, then they can find out the position of the gene on the chromosome. And this is called gene mapping, because we're actually finding the position of the gene on the chromosome, which is basically like a map of the gene mapping. So this is helpful to identify certain abnormalities that may be present, so disorders, mutations, anything like that. And there are two kinds of gene mapping. One is called linkage mapping. This deals with the location of the gene relative to other genes. So if I said that the frequency between A and B is 20, and the frequency between A and C is 50, and the frequency between C and B is 30, then the locations can be calculated as it's first A, and then B, and then gene C. So another kind of gene mapping is called physical mapping, and this is a more direct approach of gene mapping. Instead of finding genes that are relative to each other, we know the genes based on their genetic markers, or the fluorescent dyes, and then the genes are subsequently arranged, and the order is found in this way. So, also during meiosis, when daughter cells are getting chromosomes passed down to them, there are cases when they do not get the correct number of chromosomes. One of these cases is called non-disjunction. This is basically when the daughter cells is given too many chromosomes and another daughter cell is given not enough chromosomes. So this can happen in mitosis, and it is the cause of many disorders. One of them is Down syndrome, which is when there is one extra 21 chromosome 21, and Turner syndrome, when there is only one X chromosome in females. There is also an abnormality within chromosomes called aneuploidy. This occurs during fermentation, and it is when a normal male or female gamete fertilizes with an abnormal male or female gamete. In these situations, pregnancy loss is common, so the baby will not be able to be born. Finally, for our purposes at least, there is an abnormality called polyploidy. So in these disorders, in the disorders we discussed before, it kind of entails having an abnormality for just one chromosome in the set of 46 that the cell has. For instance, Down syndrome is when chromosome 21 has 3 chromosomes instead of having 2 chromosomes. But for polyploidy, extra chromosomes or multiple chromosomes are added or deleted to all 46 chromosomes. So instead of just having 3 chromosomes for chromosome 21, it will have 3 chromosomes for every single chromosome set. So this actually affects the appearance of the organisms less than just having an abnormality in one chromosome. Also, we use polyploidy to our advantage when it comes to genetically engineering plants, as when we use this on plants, we can make them bigger, as we see in these genetically modified strawberries as compared to the organic strawberries, because these strawberries have an extra set of chromosomes, which makes them bigger as they're producing more and more of each gene and protein and enzyme. So in addition to having in addition to having abnormalities which deal with the whole chromosome being removed or deleted, or like a whole set in case of polyploidy, there are also mutations that occur in chromosomes that are just a change in the genetic material itself. There are 2 categories for mutations. There are gene mutations and there are chromosomal mutations. Gene mutations involve changes in a couple of nucleotides that may affect the gene, and chromosomal mutations are changes in the chromosome structure in general. So a gene mutation can be further broken down into a couple of categories: substitution, insertion, and deletion. Substitution is when one nitrogenous base is substituted with another nitrogenous base by accident, thus changing the sequence. So, for instance, if it was AATG, then it might change. If it was substitution gene mutation, it might change to AAAG. So one of the chromosomes just got substituted. Another kind of gene mutation is insertion. Insertion is when a base is added to the DNA sequence, and this can cause disorientation within the sequence, as it has all of the bases move over one base. So basically, it's just adding one extra nucleotide that it doesn't need, and this kind of changes all the pattern and all the proteins and enzymes that are being created with this strand. There's also deletion of that's in the subcategory of gene mutations, and this is when a nucleotide is deleted, and this also causes disorientation because it's basically the same thing with, instead of one being added, one is taken away, and so this causes different amino acids to correlate with different segments of the gene, and thus creating different proteins and different enzymes. So, as for chromosomal mutations, there are 4 submutation categories. The first is deletion. So, like the gene mutation deletion, chromosomal mutation deletion is when part of the chromosome as a whole is deleted. And there is also another kind of chromosomal mutation which is called duplication. So this is when there are extra copies of the chromosome in the DNA. So it has this whole extra, basically, clump of DNA that is not needed. Kind of like insertion, but again, this is a lot bigger. So the third kind is inversion. This is when the replicated chromosomes are basically reversed. They're put in the wrong order in terms of the genes themselves. The fourth kind of chromosomal mutation is translocation. This is the only chromosomal mutation that affects 2 chromosomes, and it's when 2 sections of the chromosome swap. Now, this may sound familiar, as in crossing over that we previously discussed, but in translocation, the genes that are swapped are not the same kind of genes, so this causes many other kinds of problems. So that was it for this video. I hope you learned a lot about the different genes, recombinants, linked genes, abnormalities that we find within the genes, mutations that we find within the genes, and I hope you broadened your knowledge of genetics in general. And here are a couple of citations I used for the pictures for this video, and if you liked these kinds of genetic videos, then feel free to subscribe to my channel and like this video, and watch the other genetics video, as this is a series of videos on genetics, and there are more to come. And if you check out this channel. There's also some videos on computers, anatomy, and you can leave comments on any other academic videos you would like me to post about. I will try my best to make that happen. Again, thanks for watching. Bye.